Well, good afternoon, everyone, and welcome to the Science of Childhood Cancer lecture series hosted by the St. Jude Comprehensive Cancer Center. My name is Greg Armstrong, and on behalf of Melissa Hudson, who, along with me, is a co-leader of the Cancer Control and Survivorship Program within the St. Jude Comprehensive Cancer Center, I welcome you to this lecture. It is my great pleasure today to welcome our speaker, Dr. Lindsay Morton, Deputy Branch Chief of the Radiation Epidemiology Branch of the National Cancer Institute's Division of Cancer Epidemiology and Genetics. Before I formally introduce Lindsay, just a note that you can submit questions at any time during the talk using the chat function and sending questions to all panelists. So I'd like to also let you know that there are additional opportunities for questions beyond the top of the hour. Dr. Morton has kindly offered to stay on past the top of the hour for anyone who'd like to have further discussions. So now I'm delighted to introduce to you Dr. Morton. Lindsay received her undergraduate degree from Dartmouth College and her PhD in epidemiology from Yale University with a focus on cancer epidemiology. She then joined the National Cancer Institute's Division of Cancer Epidemiology and Genetics as a postdoctoral fellow with a concentration in molecular epidemiology. During her postdoctoral and doctoral training, she focused her research on understanding the causes of lymphoid neoplasms. When Dr. Morton then joined the DCEG's radiation epidemiology branch as a tenure-track professor, she expanded her research to include the study of multiple primary cancers, and her work has also enumerated the carcinogenic effects of radiotherapy and chemotherapy, other environmental exposures, and, importantly for this lecture, genetic risk factors for second cancers and survivors. In 2015, Dr. Morton was awarded scientific tenure by the NIH and became a senior investigator, and in 2020 she was appointed Deputy Branch Chief. She also was elected as a member of the American Epidemiological Society. So with that, Lindsay, I welcome you, and the stage is yours. Thank you.
Thanks very much, Greg. Well, I appreciate the kind introduction, and it really is an honor to have the opportunity to contribute to this terrific lecture series. I've spent many a week on the opposite side of the screen, and I'm really delighted to have the opportunity to share some of the research that you and I've been doing together. In fact, and some major collaborations over the last decade, as well as some other research looking at genetic susceptibility to subsequent neoplasms in childhood cancer survivors. So I'd like to begin and just keep in mind this perspective that I think we're all very familiar with, which is that there have been substantial improvements in survival following childhood cancer diagnosis for children who have been diagnosed and treated in successive decades from the 1970s through today. And with this improvement in survival, there's been a substantial increase in the focus on long-term health, thinking about childhood cancer survivorship. And in fact, if you just look up childhood cancer survival or survivorship in PubMed, you can see just a dramatic increase in the number of publications with each increasing year over the last several decades, representing this increased focus. One of the seminal reviews that I often turn to when I'm thinking about this field, and also when I'm introducing trainees to the field, is this review by Les Robison and Melissa Hudson in Nature Reviews Cancer from 2014, which is really talking about the lifelong responsibilities that survivors have and some of the issues that come up. And it really is this incredible breadth of different topics. And I think survivorship really encompasses this wide range. But I've chosen to focus my research on one particular outcome in this carcinogenesis box, and that's specifically the occurrence of subsequent neoplasms. I've been interested in this topic because as part of the Division of Cancer Epidemiology and Genetics, where a lot of our research has been focused on etiology, we think about the etiology of subsequent neoplasms in addition to those of first primary neoplasms. And this is also a major cause of morbidity and mortality in childhood cancer survivors. And so the opportunity to try and learn more about the causes and potential opportunities for prevention of this important outcome is a really impactful area to be working in. This figure from Lucy Turcotte's paper from the Journal of Clinical Oncology in 2019, looking at the burden of subsequent neoplasms in data from the childhood cancer survivor study that is run, that you have so ably run, Greg, for the last several years, taking over leadership from Les, really demonstrates the substantial increase in subsequent neoplasm burden and the relationship to treatment. And so what's shown here is this high risk of subsequent neoplasms for patients who were treated with both chemotherapy and radiotherapy, and also with radiotherapy in the absence of chemotherapy. The goal of this paper, of course, was to show the important role of chemotherapy alone as well. But a lot of my research focused in the radiation epidemiology branch at the in DCEG at NCI has been focused on this role of radiotherapy. Peter Inskip wrote a really nice review paper in 2016 that summarized the radiation dose response across a range of different types of subsequent neoplasms. So depending upon the type of childhood cancer and the treatment that each patient received, individuals may be at risk for different types of subsequent neoplasms. And the most commonly occurring are shown here. And what you can see is that generally there's a linear increase in risk of subsequent neoplasms occurring in all these different tissue types with increasing radiation dose of different magnitudes, the highest magnitude for sarcoma down to breast here. And then of course this outlier, which is thyroid, where the dose response in this dose range is actually not linear, but in fact has a downturn in risk with increasing doses. So if we think about kind of this broad range of subsequent neoplasms that can occur, I had the opportunity to work with a number of colleagues at the American Society of Clinical Oncology for an education book piece a few years ago, where we started to think about what is the role of genetics in adverse outcomes in childhood cancer survivors. And I, of course, have focused on the malignant outcomes because of my interest in subsequent neoplasms. So in order to think about the genetics, it's really valuable to, I think, put in perspective the incredible advances in germline cancer genetics studies that have occurred in the last several decades. So if we just think in very broad brushstrokes about our understanding of rare variants, sequencing of single genes started several decades ago, expanded to the use of gene panels, then whole exome sequencing, and now whole genome sequencing, which has really just come online in the last decade or so. And I would actually argue it's still really coming online very rapidly with expanding use with next gen sequencing and then also the bioinformatics that go along with the incredible onslaught of data that comes with sequencing. And then also the questions about how to interpret sequencing data. So we can now sequence the entire genome and look at associations with a number of health outcomes, of course, including cancer. I'm not going to spend as much time talking about this today, but I'd be remiss to not point out that there's been this incredible advance in germline cancer genetic studies of common variants at the same time. First, looking at candidate single nucleotide polymorphism. So these are individual places in the genome that are known to vary within populations. Expanding that to looking at specific genes of interest or genes that are in the same biological pathway. And then finally to looking across the entire genome at more common genetic variation. In one of the previous lectures in the series, Dr. Yutaka Yazui talked a little bit about a lot of the common variant work that's been going on. But for the purposes of today's talk, I'm going to focus on rare variants. So the way that we think about the germline genetic architecture of cancer was captured really nicely over a decade ago. And what this figure shows is the relationship between the effect size, so the amount of risk that's associated with a genetic variant, and the allele frequency, so how common it is along the x-axis. And in this bar that goes diagonally through the middle of the plot, what we really see is this is where most of the discoveries have been made. Generally, there are rare alleles that have very high effect sizes, but they don't occur very often, down to the darker brown, more common variants that occur in the population, but the risk associated with an individual variant tends to be relatively small. And then there have been very few studies that have found examples of associations that are outside of these dotted lines. So in the bottom left corner, in the white circle, the rare variants with small effect. These are just incredibly difficult to find because they are rare and the effects are small. And so the sample size that would be needed to identify them has really been difficult to attain in any studies. And then we just plain haven't seen many examples in this top right corner in the blue circle of more common variants that are associated with relatively high risks. So what I'm going to talk about today is a little bit more in these on this left side, the rare variants. And by default, what that means, especially based on the sample size, which is something a theme I'm going to come back to throughout this talk, is that the effect sizes tend to be relatively high. The risks tend to be relatively high. So when we think a little bit about genetic susceptibility to subsequent neoplasms, there are a few different models that we can think about. And it's important to consider these different models because it informs the way we approach our study design and our analytic approaches. So the first is a direct effect of a gene variant on subsequent neoplasm risk. The next is a gene variant that impacts the way childhood cancer treatments impact risk of subsequent neoplasms. So I showed in the background that the treatment-related risk for subsequent neoplasms is very well established. And one of the questions we can ask is whether a germline genetic susceptibility can alter that risk associated with treatments. And then the third is an idea that there could be a gene variant that would influence both the risk for childhood cancer and also for a subsequent neoplasm. And the reason to try and figure all of this out, if genetics really could play a role in subsequent neoplasm risk, is because of the potential for the impact. So, from a biological perspective, if we could identify gene variants that impact susceptibility to subsequent neoplasms, then we could provide insight into treatment related carcinogenesis or potentially even carcinogenesis more broadly, and also really importantly have an impact on clinical care. So there's the potential that the understanding that certain individuals might be more susceptible to, for example, a treatment-related subsequent neoplasm could impact frontline therapy decision making. I think that's relatively complicated, and there are a number of tumors where, in fact, there aren't necessarily multiple treatment options that could be selected from based on genetic susceptibility to subsequent neoplasms. So where I generally think about focusing my energy from a clinical perspective is the idea of informing long-term surveillance of cancer survivors or risk stratifying cancer survivors. If we can identify certain patients who are particularly high risk, then their long-term clinical care might look different than an individual who has lower risk. To give you a feel for the clinical follow-up guidelines and the role of genetics in those guidelines, I wanted to just highlight the Children's Oncology Group long-term follow-up and an example with breast cancer screening, where currently annual mammography with adjunct MRI is recommended after chest radiotherapy, beginning 8 years after treatment or at age 25 years, whichever occurs last. So if there's a child who's treated with chest radiotherapy at a relatively young age, say in childhood or adolescence, then they may begin twice annual screening at the age of 25, which really is a substantial burden both for the patient and for the medical system. And so if we could identify which patients are really at higher risk, for example, by germline genetic susceptibility, and others where maybe they don't have to worry quite as much, then that would have a lot of impact on patients and on clinical care. But right now, the clinical follow-up guidelines simply state the word consider, that the patient history of BRCA1, BRCA2, ATM, or TP53 mutation should be considered in this recommendation for breast cancer screening. And my hope is that someday what we're going to be able to do is actually provide a quantification of risk associated with gene variants so that there can be more guidance rather than just consideration. So I want to orient you to two studies that I think hopefully many people in the audience are very well familiar with. The two main sources for genomic studies in childhood cancer survivors come from these um, unique cohorts, really unique in the world in the sense of the availability of their data. So the first is the Childhood Cancer Survivor Study, which is in two parts, which is why there are two brackets here. The first being children who were diagnosed from 1970 to 1986. And then the next set, the expansion cohort from 1987 through 1999. And the other cohort is the St. Jude Lifetime cohort. And so that includes survivors who were treated at St. Jude over a longer time frame. And I should mention that the CCSS is a multi-center study. There are over 30 centers to date in North America. And these cohorts are unique for the fact that they collect long-term systematic follow-up for new malignancies, and they have germline DNA on a subset of participants, in addition to having detailed treatment data. And I think a number of us have searched far and wide for other study populations where this combination of data is available, and it really is just very rare. And so the ability to collaborate with you all on these cohorts is really an incredible opportunity. To give you a sense of the genomic data that's available, as I mentioned, I'm going to focus on the rare variant data. And we had the opportunity at the NCI to conduct whole exome sequencing on about 5,400 individuals from the CCSS original cohort, approximately 1,500 of whom have developed at least one subsequent neoplasm as of the last follow-up. And those data are currently available in the database of genotypes and phenotypes, or dbGaP, for download. Over the last year, CCSS has actually also made an effort to deposit their phenotype data in dbGaP, and there's an opportunity to link the genotype and the phenotype in that resource. The other source of genomic data for the CCSS expansion cohort and the St. Jude Life cohort is whole genome sequencing that was conducted by the team at St. Jude. And I provide also here the counts for the total numbers, about 2,900 in the CCSS expansion, approximately 200 of whom have developed at least one subsequent neoplasm, and about 4,600 in St. Jude Life, 440 of whom have developed a subsequent neoplasm. And I'll talk a little bit about why there's an approximate here, because there's some overlap of individuals in the different studies. But those data are all available on the St. Jude Cloud, which we began working with a couple of years ago, and I'll talk more about that later in this lecture. So let's get to some data. So when we think about genetic susceptibility to subsequent neoplasms, one of the first things to look at or to think about is, what about known cancer susceptibility genes? So there have been a couple of analyses on this topic that have already been conducted. The first is to actually just look at the frequency of pathogenic or likely pathogenic variants, so rare variants that are thought to be damaging to the protein in known cancer susceptibility genes. And that frequency was estimated in an analysis led by Jung Kim, actually at the NCI, published in 2021. And that number was about 4.1% in CCSS, as opposed to about 1% in controls. And I highlight here just a few genes that were specifically emphasized in this paper, and for specific types of childhood cancer. So this is not the subsequent neoplasms, but the childhood cancers themselves. And what you start to see here is, of course, known cancer susceptibility genes, and then you start to actually see also some genes that are associated with DNA repair, which is another topic that I'm going to spend time on next. So that was in the CCSS. What about the St. Jude Life? Xiaoming Wang published this paper in JCO in 2018, looking at pathogenic or likely pathogenic variants also in St. Jude Life, and came up with a similar number. Nearly 6% of individuals in St. Jude Life carried pathogenic or likely pathogenic variant in one of these genes. And you can see here the distribution, now not by the first cancer type, but actually by the subsequent neoplasm type. And again, a lot of the key genes that you would expect to see associated, for example, BRCA1 and 2 and TP53 with breast cancer, are highlighted here. But you can also see in parentheses the relatively small numbers of individuals who are identified with these rare variants. And one of the things that has really been a question was whether subsequent neoplasms were predominantly occurring among childhood cancer survivors who had variants in these known cancer susceptibility genes. And what we can see in the long-term survivor cohorts in both CCSS and St. Jude Life is that that answer is no, that actually the fraction of individuals is less than 10%. And so with the much higher cumulative incidence of subsequent neoplasms than that, these known cancer susceptibility genes can't account for a major fraction of the subsequent neoplasms in these populations. So looking a little bit further, again, with the data from St. Jude Life that was published several years ago, this is looking at the frequency of having at least one pathogenic or likely pathogenic variant in a known cancer susceptibility gene versus not, separately for irradiated survivors and then for non-irradiated survivors. And this figure really nicely shows that in irradiated survivors, the elevated risks really were for subsequent breast cancer and sarcomas, whereas for non-irradiated survivors, it really appeared to be actually for all outcomes. So this is starting to get at that question that I was raising about the potential for a genetic variant to directly influence subsequent neoplasm risk. And that's what's shown in this bottom plot in the non-irradiated survivors, as opposed to kind of a joint effect of the gene variant and the treatment on subsequent neoplasm risk. And that's what's shown here in this blue figure with the irradiated survivors. So moving on from the known cancer susceptibility genes, what about DNA repair genes? So it's long been known that radiotherapy and chemotherapy are cytotoxic and that the cellular damage needs to be repaired. And so the question is whether variants in DNA repair genes could impact subsequent neoplasm risk. And so we undertook an analysis in the CCSS original cohort with the whole exome sequencing data that we had and looked at a number of the key DNA repair pathways, double strand break repair, single strand break repair, et cetera, and also some of the radiation sensitivity genes. So the variants in genes that are known to confer clinical susceptibility to adverse effects of radiotherapy. The primary outcome was the occurrence of any radiation-related subsequent neoplasm type. And as I mentioned, because the children in the childhood cancer survivor study original cohort were diagnosed a longer time ago and have been followed for a longer period of time, there's a substantial number of individuals who've developed a subsequent neoplasm. In this analysis, it was just over 1,100 individuals. And we did these analyses overall by the specific subsequent neoplasm type, and also considering the dose to the subsequent neoplasm location. So again, we're starting to bring in this question of could there be a dose-specific effect on radiation-related risk and the impact of genetic susceptibility based on that exposure. And so this figure shows in the red bars the subsequent neoplasms that essentially occurred in the radiotherapy field. So based on the dose reconstruction from the radiation physics team at MD Anderson, now led by Rebecca Howell. The majority of the subsequent neoplasms we know do occur in field, so they're exposed to at least 10 gray. In the light blue, those are the out of field tumors, and so they were exposed to less than 10 gray of ionizing radiation. And then in black, in the unirradiated survivors. So I'd like to highlight some of our top results, and this was to identify DNA repair genes in CCSS, specifically variants in the homologous recombination pathway. And what we saw, I can orient you here, to first the frequency of carriers in the cases compared with the frequency of carriers in the controls, the odds ratio, and then the p-value. And what we found was about twofold higher risk of developing an out of field subsequent neoplasm among individuals who carried variants in homologous recombination genes. When we looked at these outcomes specifically for different types of subsequent neoplasms, we actually saw relatively consistent risks. In other words, odds ratios above 1 for all subsequent neoplasm types, although this only reached statistical significance for the breast cancer patients. And then when we tried to look at individual genes, there are actually 87 genes in the homologous recombination pathway. There were 16 genes where there was at least one case who carried a variant and had an out-of-field tumor, and 7 genes where there were at least 2 cases. And I show the results for these 7 genes here. And what you can see is a higher frequency of carriers in the cases for developing any type of subsequent neoplasm compared with the controls. And again, odds ratios all above 1. But I think it's really important, and we've tried throughout to show the sample size when we're looking at these rare variant analyses, and that these findings for individual genes are very limited based on small sample sizes. And so it's really important that we interpret these results cautiously. I want to pause for a moment. I'm going to present results from a few different collaborative efforts, and just want to make very clear that all of this really represents work by a tremendous interdisciplinary team of investigators. And I often say it takes a minimum of 4 PhDs and a couple of MDs in order to do all of this research, which obviously no one individual can have. And so I really appreciate the terrific collaboration that I have with so many people who are trained in different disciplines and bring different expertise in order to carry out these studies. So turning now to DNA repair genes also in St. Jude Life. So the data that I previously showed were the whole exome sequencing data from the CCSS original cohort, and now looking at the whole genome sequence data from St. Jude Life. They also saw suggestive increased risk of subsequent neoplasms, particularly in individuals who carried pathogenic or likely pathogenic variants in homologous recombination genes. This figure shows the results. This is a paper from JCO in 2020. Um, and this figure shows the results for breast cancer overall, and then also stratified by treatment exposures. And what you can see is the increased risk, particularly evident for the children who received chest radiotherapy, at least 20 gray, and also higher dose anthracyclines. They also found increased risk for subsequent sarcoma occurring among individuals with pathogenic or likely pathogenic variants in homologous recombination genes, particularly, so overall, and then particularly individuals with higher doses of alkylating agents. But I think it's really important, again, to think a little bit about the sample size. And so this paper is terrific in the sense that all of the data are available. And in the supplement, you can actually see the counts of the numbers of survivors, the numbers of survivors with mutations, and then therefore the mutation prevalence. And what you can see is that the results for homologous recombination specifically for breast cancer and sarcoma are based on 7 cases for breast cancer and 4 cases for sarcoma. So it again really illustrates, just as I mentioned for the analysis that we published from the CCSS original cohort, some of the challenges that we have in looking at rare variants. Although the relative risks are high, because they're rare, the sample sizes are very, very low. So we have an ongoing pooled analysis of DNA repair genes, and I want to bring in just a little bit of a feel in thinking about kind of the general approach to cancer genomics and bioinformatics and how this is really playing out in these survivor populations. So I mentioned that we have the CCSS original cohort whole exome sequencing that we conducted at the NCI with the data posted to dbGaP. And we had called the variants to identify the pathogenic and the likely pathogenic variants using a pipeline of multiple callers, which increases the reliability of the variant calls. It's a little bit trickier to identify variants from sequencing data than, for example, identifying genotypes from SNP array data. And some of the callers that we used actually were no longer supported because they had been updated. And so over the last couple of years, we have updated the pipeline with new callers in order to identify the high quality variants in the whole exome sequencing data. We also have appreciated the opportunity to access the whole genome sequence data from the St. Jude Cloud. I think that there's a tremendous amount of promise in cloud computing, and there's a lot of discussion about the idea of taking the code to the data in the cloud rather than having many copies of the data, particularly when you're talking about hundreds of terabytes of data as we're talking about here. But the reality of that is very complicated still. And there's a lot that the field needs to do, in my opinion, in order to make cloud computing for genomic studies a reality. Because the computational costs, for example, for running multiple callers, potentially realigning sequencing data, is really infeasible in my mind. So we estimated what it would cost us to try and run these callers in the cloud. And when our bioinformatician got to about $750,000, I said, you can go ahead and stop. We don't need any more exact data. We're going to go ahead, and we collaborated with our chief data scientist in our division. And actually we sliced the whole genome data to the whole exome data, which is a very small fraction of the genome, and have a copy of that locally, realigned and called the variants with this uniform pipeline so that we can have a harmonized data set across all of the available data for these childhood cancer survivor cohorts. At the same time, we've been updating the subsequent neoplasm, the treatment, and the follow-up data from the cohorts. So I mentioned the CCSS original and expansion data are in dbGaP with updated outcomes compared to our previous analysis. And so we're using those updated data. And we really appreciate the St. Jude Life team as willingness to share their phenotype data, and just within the last couple of months have shared phenotype data for subsequent neoplasms from the St. Jude Life. And so we're really excited about this ongoing pooled analysis that will bring together this fully harmonized data set on both the genotype side and the phenotype side to look at DNA repair genes and subsequent neoplasm risks. So this has been an incredible effort, as you can imagine, to actually do all of this. And so I want to take a slight detour and share some parallel work that I've been doing where we've really been focused on DNA repair. And it really has helped motivate our team, I would say, for doing the germline genetics work, is actually to do some somatic work with a very unusual opportunity that we had to do a comprehensive molecular characterization of thyroid tumors that occurred after the Chernobyl nuclear power plant accident. And this was a paper that we had the opportunity to publish in Science about a year ago. So many of you may recall that in April 26, 1986, there was an explosion in one of the reactors at the Chernobyl nuclear power plant. And that explosion and the resulting fire that was only contained about 9 days later dropped radioactive contaminated fallout in the surrounding regions. And in those pastures, there were cows that were grazing as well as leafy greens that were available. And decades of epidemiologic research in collaboration with Ukrainian, Belarusian, and Russian scientists have demonstrated that one of the most important adverse health outcomes from the Chernobyl nuclear power plant accident was the occurrence of thyroid cancer. That was particularly evident in children who had consumed dairy products from cows who had grazed in the contaminated pastures, or also children who had received or consumed contaminated leafy greens. So what we were able to do was to partner with some visionary scientists from the Chernobyl Tissue Bank who began collecting fresh frozen tumors from individuals who were diagnosed in Ukraine with thyroid cancer beginning in the late 1990s. And this Chernobyl Tissue Bank is currently led out of Imperial College London by Jerry Thomas. And she and Ukrainian colleagues collected these thyroid tumors, and with the understanding or the hope, the promise of the advances in genomics and the idea that one day we would actually be able to undertake an analysis like this. And so what we did was a cancer genome-like analysis where, with whole genome sequencing, RNA-seq, methylation profiling, we actually did a whole genomic landscape analysis and then related all of the molecular characteristics to the radiation dose. And what we were able to show was an increasing number of DNA double-strand breaks in the tumors themselves with increasing radiation dose. So while it's been shown for decades and radiobiological studies that DNA double-strand breaks are one of the most important forms of radiation-related damage, it's never actually been shown to be the mechanism of carcinogenesis itself. So I want to give you a little bit more of a feel for some of the sub-analyses and why I feel like this is really important work that marries with the germline work that I've been doing with collaborators in the childhood cancer survivor cohorts. So this is a really nice review paper that was published in 2017 that talks about the key DNA double-strand break repair mechanisms: non-homologous end joining, alt end joining, and homologous recombination listed here at the bottom. And the key is, we've been talking about homologous recombination, which is a very high fidelity repair. Because the opposite strand is available when there's damage to the DNA, with homologous recombination, essentially an exact repair can be undertaken. So it's considered very high fidelity. In contrast, with non-homologous end joining, there's a small amount of DNA that's actually removed. There's a small deletion, essentially, that's cut out, and then the double-strand DNA are repaired and brought together. And so different repair mechanisms can generate deletions of different lengths, which means we can see the scars of certain repair mechanisms in the DNA of the tumor itself. Whereas, of course, with homologous recombination, because it is such high fidelity recombination, we can't actually see any evidence of the homologous recombination activity. You also can look at the microhomology between the DNA ends, and it turns out that the amount of microhomology also reflects the DNA double-strand break repair mechanism. And so we can use these pieces of information, and we did use these pieces of information when we profiled the thyroid tumors to actually see how the double-strand breaks were repaired. So what we did was we identified the deletions and we focused on those that are at least 5 base pairs long, because that would be enriched for end joining repair. And then we characterized all of the deletions by the amount of microhomology at the deletion boundary, with the idea that if there's 0 to 1 base pairs of microhomology, then it's enriched for non-homologous end joining, as opposed to 2+ base pairs, which is a mixture of non-homologous end joining and theta-mediated end joining, or an alt end joining mechanism. And what we were able to see, so I mentioned that what we did was we were able to relate the characteristics to the radiation dose. And what we were able to see was an increase in the number of 5 base pair deletions associated with increasing radiation dose. And that finding was consistent regardless of the amount of microhomology. And so if we go back here to the microhomology slide, what we can see is non-homologous end joining is evident throughout this range of microhomology, whereas alt end joining is really only evident with several base pairs. And so the consistency of these findings, the association regardless of the amount of microhomology, implicated non-homologous end joining as a key DNA double-strand break repair mechanism, as opposed to alt end joining. So again, this was a tremendous collaborative team that carried out this Chernobyl research. A number of the key collaborators here, including Stephen Chanik, Danielle Cariati, who's a geneticist, and Steve Hartley, who's a bioinformatician, overlap between this project and the work that we've been doing in childhood cancer survivor cohorts. And so it's been really terrific to have the synergy of the teams thinking about DNA repair. So going back to known cancer susceptibility genes, the last thing that I wanted to talk about is some ongoing work that focuses more on this bottom model here, the idea of the potential for a gene variant to impact both childhood cancer occurrence and subsequent neoplasm occurrence. And when I think about this, I think of 2 main genes that have been long known to confer risk for childhood cancer, retinoblastoma 1, RB1, and TP53 with Leigh-Fraumeni syndrome. And these genes are also known to have pleiotropic effects, so they also are known to impact risk for a number of other cancers that occur later in life. What we found in CCSS and St. Jude Life is actually a relatively small fraction of individuals in those cohorts have variants in these genes. And so it's actually important to turn to other study settings in order to understand the role of these genes in subsequent neoplasm risk. So at the NCI, we've actually had a long-standing retrospective cohort study of over 2,000 children who were diagnosed with retinoblastoma over much of the last century. This was led by Ruth Kleinerman and David Abramson at Memorial Sloan Kettering, and now Joanna Seddon in Massachusetts. And what this figure shows is the increased risk of second cancers among retinoblastoma survivors. So just as I was showing you in the background in the beginning of across all childhood cancer survivors, retinoblastoma survivors have very strikingly elevated risks of developing subsequent neoplasms. And those risks are highest in individuals who had bilateral disease and have a family history of retinoblastoma. Interestingly, a little bit lower among children with bilateral disease, so a germline RB1 mutation, but no or unknown family history, and then lower with relatively small numbers among individuals with unilateral disease. So this figure is suggestive that there is some genetic effect in the impact of, there's some genetic effect on subsequent neoplasm risk. So this is a broader sense of the specific types of subsequent neoplasms. Retinoblastoma survivors face the highest risk for soft tissue sarcomas and bone sarcomas, also for melanomas. Um, and this risk varies depending upon where the tumor occurs. So in blue here, we show head and neck or in-field sarcomas for the retinoblastoma patients who were treated with radiotherapy, as opposed to those that occur out of field. And the way that we think about these risks for both bone and soft tissue sarcomas, where we see similar findings, is that the infield tumors essentially represent the combination of a genetic risk from the germline RB1 mutation plus the treatment exposure, the radiotherapy for retinoblastoma, because this cohort study includes many children in an era when radiotherapy was frequently used in retinoblastoma treatment. As opposed to the red line, the out of field tumors, and those essentially represent the genetic risk alone without the treatment exposure. And so one of the questions has been almost taking an opposite extreme approach to some of the discovery work that we've been doing in CCSS and the St. Jude team has been doing in St. Jude Life, which is to look across all pathogenic variants and likely pathogenic variants combined, even across multiple genes within a pathway. This is getting at a much finer detail. And the question is, can we look at the specific type of RB1 mutation and see whether or not there's a genotype-phenotype correlation, so a relationship between the RB1 mutation, either type or location, and the specific type of subsequent neoplasm. So a group of investigators from the Netherlands published this paper about a decade ago, and they recognized the substantial challenges in doing this with just 44 patients who had developed a subsequent neoplasm. And so for this and other reasons, the Dutch group actually has led the establishment of an international collaboration focused on RB survivorship. It's called IRISC, the International Retinoblastoma and Second Cancers Consortium. And this broad group of interdisciplinary investigators from around the world is bringing data together that's all going to be harmonized by the group in the Netherlands to evaluate treatment-related subsequent neoplasms, evaluate genetic susceptibility to subsequent neoplasms. So really building on this question of whether there are specific RB1 mutations that confer differential subsequent neoplasm risk, and then also contribute to survivorship guidelines for this high-risk population. And again, this is a terrific group of collaborators, so I want to acknowledge our team at the NCI, Memorial Sloan Kettering and Massachusetts for the NCI cohort. And then there are many individuals who are involved in IRISC, but I wanted to highlight the Dutch group that has established the consortium. And we're really excited at the NCI to be able to contribute our data to this effort under the Dutch group's leadership. I also want to turn to TP53 because of its important role in increased risk for childhood cancer and also for a number of cancers that occur later in life. So there also is a cohort of TP53 mutation carriers that's at the NCI. It's led by the clinical genetics branch, originally by Joe Fraumeni, and then the study was substantially expanded by our current chief of the clinical genetics branch, Sharon Savage, and one of our earlier career investigators, Payal Kincha. But in this analysis of 286 carriers of pathogenic or likely pathogenic mutations in TP53, what you can see is the cumulative incidence of all of these different types of cancer by age in females and then in males. And then a subsequent analysis that shows the 280, in this 286 carriers, the time from the first cancer to the occurrence of second cancer. So this is the percent of individuals who are second cancer free. So you can essentially think of this as an inverse cumulative incidence curve. And so you can see the onset of second cancers relatively rapidly for the first decade after a first cancer in both females and in males. An additional analysis that was just published in 2021 with a larger set of individuals from the NCI study again by investigators in our clinical genetics branch, included 480 carriers of individuals with pathogenic or likely pathogenic mutations in TP53. And just like the retinoblastoma story, what Kelvin DeAndrade and colleagues did in this paper was they classified the TP53 mutations by their type, dominant negative, loss of function, and looked at the frequency of cancer occurrence. And what you can see is the highest frequency in the orange and the blue in individuals who have loss of function mutations in TP53. So this again kind of is suggestive evidence that there is a genotype phenotype correlation, and that it's a good start to look broadly at the type at the existence of a TP53 mutation, but looking at the type also can provide valuable information clinically. Again, there's an international collaboration, the Leaf Romani Exploration Consortium, or the LIFE Consortium, that has been established to do a tremendous amount of collaborative work from investigators around the world who have cohorts of individuals who are germline TP53 mutation carriers. And I feel fortunate to have the opportunity to work with this consortium, particularly on a project that was initiated under the leadership of Anita Villani at Sick Kids, that's looking at treatment-related subsequent neoplasms in germline TP53 mutations. And right now, the initial kind of pilot part of this study includes patients who are contributed from the 5 centers listed here: Sick Kids, the NCI study, the St. Jude's study, a group in Germany, and a group in Utah. And we're just beginning those analyses. But again, it shows the power of the collaboration and the pooled analysis to learn more about TP53 related subsequent neoplasm risks, because we have found that we actually can't learn a tremendous amount from CCSS or St. Jude Life about this high risk population. So with that, I just want to close. I hope we can have a spirited discussion about genetic susceptibility to subsequent neoplasms, but I want to close with information about training at the NCI. We have a range of fellowships for postdocs, pre-docs, and post-bacs, and encourage you to take a look at this website if you would be interested. And I look forward to your questions.
Great. Thank you, Lindsay. What a fantastic lecture. Thank you for joining us for this lecture series. So I guess I get the privilege of asking the first question, although we have a number of questions lining up here, so this will be a good time. For me, I think about risk prediction models, and within our field, whether it's subsequent neoplasms or cardiac outcomes, we have a number of risk prediction models that have even been validated, but they really include only clinical variables, and treatment variables. When will we see the day that we have integration of some of these genetic findings into these risk prediction models? What's it going to take to really get there?
Yeah, you've touched on such an important question, and one that uh, I honestly, it keeps me up at night. So really, we have learned so much from general population cancer genomic studies and study design, laboratory approaches, bioinformatics. And one of the things that we have really found is the importance of independent replication and large sample size in order to accurately quantify risks. And one of the challenges that I really think we face in these genomic studies in survivors is that, as I mentioned early in the talk, there are very few settings in which we have um, very detailed treatment data, long-term follow-up, and DNA. And so it limits our opportunities for replication. And so what we find is the idea that you're doing discovery and risk prediction then in the same study set, which leads to the possibility that you have spurious findings. And so I think we have to really grapple with this as part of our clinical translation. And I think that on the one hand, I think we can't wait for the data to be perfect, particularly when we have some signal that there are certain survivors that have high risks. And if we can improve their clinical care, then we really want to do that. On the other hand, I think we want to make sure that we're making these decisions based on reliable data.
That's great. So, just sort of following up on that, then, do you think the effect sizes you're seeing with the genetic predisposition compared to the effect sizes just from radiation injury and chemotherapy, will those from therapy just be so large that genetics may not have an impact, or are we going to see that when we put these together, there's impact from all of them?
Yeah, we've long wondered that too. And actually, one of the analyses that I didn't describe at all today because I decided to focus on rare variants is actually going to be presented by Todd Gibson at ASCO. And so what he's done is actually looked at the polygenic risk score. So this combines the individual relatively small risks from common genetic variants and adds them together into a single risk score. And then what he's doing is looking at the joint effect of that risk score with treatment. And I think that they're really important questions. If it turns out that there is simply an additive effect, then it could be exactly as you say, Greg, that the treatment related risks are so high that really is the most important thing to focus on. And as we think about long term clinical care guidelines, for example, the current guidelines that are focused on treatment would then be adequate. But what if it's more than additive? What if it even reaches multiplicativity? Then I think you can start to imagine that you could get that risk stratification depending upon what that relationship looks like. And Todd's really trying to tackle that and is excited to present that at ASCO.
Yes, and I'll just comment that's an oral presentation at ASCO, so
It
congratulations to
is.
both of you. We'll look forward to it. So, your thyroid work in the Chernobyl population is fantastic and really gives insights into the somatic tissue findings. But when we have the opportunity here in the near future to look at somatic tissue from second cancers from children treated for their primary cancer, what do you think we're going to find? What insights would you carry forward, but what differences might we see?
I think that there have been some really exciting data that actually suggest that there could be some real consistency in the somatic changes in tumors, regardless of the radiation exposure. I think that evidence comes from actually the first, I think, really strong paper on this topic was published by Bijadi et al. In Nature Medicine in 2016, I believe it was. And there they looked at just about a dozen subsequent neoplasms that had occurred in field, but they were subsequent neoplasms of a number of different types, and they didn't have radiation dose data. But that paper suggested an excess of small deletions. Um, right after our paper came out in Science, there was a paper that came out in Nature Genetics actually looking at brain tumors that also saw an excess of small deletions after radiation exposure. But I think that there are a lot of questions. I've worried about the generalizability of our findings from Chernobyl to childhood cancer survivors, or more relevantly, I would say to even more broadly to other ionizing radiation exposed populations, because um, it's quite different to have radioactive iodine as opposed to external beam therapy. The dose ranges are quite different. The fractionation is different. And so I think the preliminary data suggests that we'll see consistency across tumor types and across dose ranges, but I think that that's really actually would be a little bit inconsistent with the radiobiological data, which suggest that in fact there are different biological responses to different doses.
Okay, well, it should be very exciting. So, we've got a number of good questions here. So, let's start with this one. Although the sample size could be limited, did you consider exploring association of pathogenic and likely pathogenic variants with subsequent neoplasms by ancestry?
One of the challenges that I think we have right now in our survivor cohorts is that we're so limited by having just a small number of studies with available data. And what we know from the broader cancer genomics field is that there's a bias, and the initial study populations are predominantly non Hispanic Caucasian. And that's where that is the case also in CCSS and St. Jude Life. But more broadly, what we've seen in cancer genomics is that there are really important insights in terms of the biology, and also really important insights in terms of clinical care when we diversify the ancestry of the study populations. And I think that's something that we're all very aware of and really need to drive towards. And when we think about expanding studies in the future,
Absolutely. It's got to be a top priority. Thank you.
it has to be.
Here's another question. So, do you think the use of polygenic risks of common variants will be important in considering a risk for second neoplasms, or will the greatest impact be restricted to high risk rare variants?
I don't think we know the answer to that yet, but I really encourage you, if you didn't have a chance to do it, to watch the lecture previously in the series by Dr. Yutaka Yazui, because he did a really nice job talking about polygenic risk scores and some of the research to date, and that's available in the archive on the St. Jude website. So I think that I think it depends what you mean by important. Certainly, the risks that we're seeing with the rare variants tend to be higher, although we're really only have the statistical power to find those higher risks. And they're only relevant because they're rare to a smaller number of individuals. And so the polygenic risk scores will inevitably have an impact for a broader portion of the population, even if that impact for a given individual is smaller. So I think we need to be pursuing both avenues. Utaka talks about a nice paper by Cindy M, where she looks at the generalizability of polygenic risk scores across a number of phenotypes. So I mentioned early on that my interests and research really are focused on subsequent neoplasms, but childhood cancer survivors face a whole range of adverse outcomes. And Cindy's paper actually looks across a range of other outcomes like obesity and cardiovascular disease.
Right. I'm going to give you one more question before we hit the top of the hour, and I guess this is really the all important or most important question is when will we ultimately start translating genetic findings into the clinical setting? I mean, you talked about risk prediction models, but when will we really get to the point that we're comfortable stratifying follow-up or even directing primary therapy based on these findings?
I think that actually some of the most exciting and promising work has come out of the TP53, the Leaf Ramani Consortium. So that's a really good example where a number of investigators all had screening protocols in that high risk population for both first and subsequent malignancies. And those individuals banded together and um, coordinated some of their protocols and their data analysis. And so I think the more that we can bring data together, the faster we'll be in terms of the clinical translation. And that promise has been shown again and again in many different settings, which is really exciting.
Good. Well, thank you, Lindsay. We are at the
Thanks
top
for
of the
having me.
hours. Yeah, so I want to thank you for a great talk and for everyone who joined us today. At this time, I'd like to invite anyone who's interested to stay on because Lindsay has been kind enough to stay on for extended discussion, and we do have a number of additional questions. So before we jump to that discussion, I just want to remind you that you can join us again on May 19th for our final lecture of the season, and we'll have our own Dr. Kaladimos presenting on the conformational landscape of protein kinases in physiology and disease. So Lindsay, thank you for staying on. We're going to dig into a few more questions. Let's see. How about this one? This sort of overlaps with something we discussed a few minutes ago, but it may give you the chance to go deeper. Have you considered examining both germline and somatic pathogenic and likely pathogenic variants with respect to subsequent neoplasms?
Yeah, so I think this would actually be a great opportunity, Greg, for you to talk a little bit more about the work that you're doing in CCSS.
Well, we have had the opportunity, I mean, really, with the wisdom of Les Robison 20 years ago, to begin to collect the somatic tissue for many of our second cancers. So over the years, we've accumulated a biobank with over 600 subsequent malignancy tissues. So ongoing somatic sequencing, whole genome and whole exome sequencing is moving forward along with RNA-seq on those somatic tissues with paired germline sequencing as well. And the sequencing is still ongoing. So I think the questions are there, the answers are not. So I think the future is open to that, and look forward to obviously making that a publicly available resource through CCSS. And yeah, so there's no answers, but there's good questions, I think.
Yeah, and to just build on that, I think I've mentioned it different times. I feel like the survivor studies have really benefited from the tremendous advances that have been made more broadly in cancer genetics. And I think one of the things that we're all really counting on is the promise that has not yet been realized. I think of some of the advances in technologies for using archival tissues. And so I mentioned that the first set of tumors that we used from the Chernobyl tissue bank were all fresh frozen samples.
Hmm.
Um, and it's just very unusual. It has to do with the fact that the thyroid tumors in Ukraine were all diagnosed centrally. under a group led by academician Mikola Tronco, and then the pathologist Tanya Bogdavina, and they were able to kind of to direct the collection of those fresh frozen tumors. There are a lot of limitations to using formalin-fixed paraffin-embedded tissues, and we're actually doing follow-up in the Chernobyl tissue bank with those archival tissues. And I think, experiencing some of the challenges that you're experiencing with FFPE tissues and CCSS.
Yep. So here's a good one, actually. This opens up a whole potential topic. Is there evidence to suggest that research directed towards methylation, proteome, metabolome, so the omics, extending this into omics will provide important insights related to subsequent cancer risk?
I think that there's a lot that can be done there. I think that that's another arena in which, there's a lot of hope and promise that has not yet really been realized. I think if we can take advantage of some of the bioinformatics and lessons learned more broadly uh, in kind of general population sporadic tumors, then we may be able to make some advances in survivor populations as well, but not yet.
Very specific question, Lindsay. This is the question there are more than 50 types of soft tissue sarcomas. Is there a hazard in lumping them together? And I guess the real underlying question is, did your studies lump all soft tissue sarcomas together?
Yeah, I appreciate that, and I actually, I really appreciate the use of the word lump, which I know sounds kind of funny,
I'm
but very
quoting.
no, it's exactly no, it's the what's in the chat, but I really appreciate the use of that word because early in my career, where I had the opportunity to begin studying hematologic malignancies, there was a question that was posed, the possibility that there would be etiologic differences in different types of hematologic malignancies, specifically different types of non-Hodgkin lymphoma. And in the International Lymphoma Epidemiology Consortium, we really engaged in this dialogue, which boiled in part down to, are you a lumper or are you a splitter? Do you believe that you should group the different tumor types together, for example? Maybe they have some similarity in characteristic that could justify them being lumped together, like the soft tissue sarcomas, that they all occur in the same tissue type. Um, or do you fundamentally believe that actually there are key differences and that by lumping them together, you're essentially introducing misclassification and reducing your statistical power? So do you increase your statistical power by increasing your sample size, or do you reduce it potentially by increasing misclassification? So this has been something that I've grappled with through my whole career, and I tried to introduce it, although I didn't use the word lump in the actual presentation, but I tried to introduce it in the concepts of looking at both the RB1 mutation and the TP53 mutation. So I think that there is good evidence that there is genetic variability in risk for different soft tissue sarcomas. So to directly answer the question, I think that it could be a limitation
Mhm.
for certain variants to lump them together. We don't have the statistical power to separate them. So what we would have to be able to find would be a gene variant or a gene with pathogenic variants that conferred risk more universally across all or a large subset of soft tissue sarcomas. But I think it's a fundamental question of study design that we will continue to grapple with.
Right, no doubt. So here's one in a different direction. So, what laboratory follow-up is underway to investigate how identified genetic variants can facilitate transformation of normal cells in the presence of ionizing radiation? So, I guess we're really getting at functional studies and
Yeah.
mechanism. How are we going to really learn beyond what we've done?
That's a great question. And in partnership with a number of different geneticists, I've had some really interesting conversations, more focused on trying to answer the question of when. When is an association study finding strong enough that it is worth the investment, the substantial investment of resources to do those functional studies? And that's a judgment call that I think we still struggle with in certain times. On the one hand, the functional work can actually support the discovery work and support the biological plausibility, or actually be inconsistent. So that would suggest that perhaps your discovery is a false positive finding. but it really is a significant investment of resources.
Yeah.
Um,
So here's a really specific question, and don't want this to put you on the spot. It's not exactly from your lecture per se, but do we know the genes for subsequent neoplasm risk for GBM, glioblastoma multiforme?
we don't. It's a relatively rare group, and based on the sample sizes, we haven't identified specific gene or gene variants to date.
Right, here's another that's fairly specific and but it's a good question here. Are there differences in outcomes and clinical features of secondary tumors compared to the same tumor types that are non-secondary, where they're primary cancers?
Yeah. So I also appreciate this terminology issue. So the concept of a secondary cancer, I've always interpreted as one that attributes causality. And so it's, for example, secondary to the radiation exposure. One of the interesting things that I didn't emphasize during the main talk about the Chernobyl findings was that we did not find a unique biomarker for radiation exposure. So we cannot differentiate those tumors that were for sure caused by radiation because we could not identify that unique signature. And so it becomes in some ways, a statistical question about a second primary tumor as opposed to perhaps a first primary tumor, but not using that word secondary because we actually cannot specifically know those tumors that were caused by treatment. There have been a few analyses that have tried to simply ask that question: what do the outcomes look like for second primary tumors, as opposed to first primary tumors controlling for key patient factors like age at diagnosis and treatment. And I would say the evidence to date is limited and a bit mixed in terms of the outcomes and clinical features.
Right, and the one that comes to mind, Haya Moskowitz had a really nice paper just a
Yeah.
couple of years ago in JCO, looking at mortality after breast cancer and compared breast cancer among childhood cancer survivors to that in SEER, and identified that the mortality rates are higher, all cause, but when she actually dug into the reasons for mortality in the survivors, a lot of it was due to other morbidities, and so this compounding of multiple morbidities that the breast cancer then just tipped them over the edge. So it's certainly a complex question and a complex answers. So great question. Um, so given our understanding of the contributions of cancer treatment exposures to subsequent neoplasm risk, how do you expect the genetic etiology of specific SNNs to differ from the genetic etiology of corresponding cancers that are primarily seen among adults? Again, overlapping with some of the previous questions, but
Yeah, so I kind of frame that as asking this question of generalizability. So it kind of goes both ways. Will we find that the variants discovered in the general population are also relevant for survivors? And so far, the evidence suggests that they are. The magnitude maybe of risk may be slightly attenuated, but they're still relevant in survivor populations. Um, and then I think most of the survivor specific findings really have been based on particular treatment exposures. And so you wouldn't expect those to generalize to sporadic cancers in the general population. And indeed, that's what we see so far.
okay, we have one last question, Lindsay. Are you ready?
I'm ready.
Did great. Okay, this one, it's the same one I asked you, talk. It's about survival bias and what's the impact of survivor bias on some of these works. We know in much of our research, there's no impact, and in others, there's a lot of impact. So just like to get your thoughts on how you consider and think about survivor bias in the scheme of your own work.
Yeah, so actually, in the paper that we collaborated on, Greg, as you know, in the supplement, the DNA repair paper, that's JCO Precision Oncology in 2021. There's in the supplement, there's actually a figure that shows the availability of DNA by time since childhood cancer diagnosis, and then patient's vital status. And what we can show is, of course, exactly what you would expect, that because the study was started in the 1990s when the children were originally diagnosed in the 1970s, then and the biospecimen collection was a few years after that. That incompleteness introduces a survivor bias. um, it's interesting because one of the things that we showed in our first collaborative paper looking at the breast cancer GWAS was that actually a previously published SNP in PRDM1 had been associated with subsequent neoplasm risk, but actually appeared to be that was in a study designed with super controls. So the controls all had to be alive much longer than the time period from the case's childhood cancer to their subsequent neoplasm development. And that SNP appeared to be associated with survival. That gave us a bit of a scare, I have to be honest. It was very sobering because in the general population, there are very few variants that have been identified to date that are associated with both susceptibility and survival. Um, I think increasingly we are finding those variants, and so thinking about the survival bias is really important. At least for RB1 and TP53, I'm hopeful that we will be able to use some of those consortial efforts to get at survival bias. But they actually the contributing studies in those consortia have some different biases in terms of the data collection. So I really think when we think about clinical translation and the ultimate impact of this work, we have to put the puzzle pieces together from a number of different sources to really figure out what's going on.
Well, Lindsay, thank you so much. A great lecture, great questions from the
Thanks.
audience, and great answers. And it's a credit to all that's been done, but it also really illuminates the future and where we need to go, what we need to do. So many things to tackle. So it's been great to collaborate with you, not only in this lecture, but in some of the research. So thank you so much. And we will remind everyone, we'll see you again in 1 week for our final lecture of the series. Take care, everybody.
Thank you.
Bye-bye.